And now, a few common examples. It is not a spore former, so we don't have to worry about those really heat-resistant seed-like structures. There are over 2,000 different species or serotypes known, many of them identified by where they were initially isolated. It is commonly found in the intestinal tract of warm-blooded animals, humans, insects, a variety of different creatures, but it can also occur very well in the environment, and it survives freezing and dehydration extremely well. Keeping this organism in check can include thorough cooking or pasteurization of a product, refrigeration to keep it at a nice cold temperature so it's not able to grow, a low pH. Once the acidity is high enough or the pH low enough, the organism should not be able to grow. And I would also add to that the low water activity will keep it from growing. It may not kill the organism, but it will keep it from growing. Also proper hygiene, both in terms of personal hygiene of the people operating the facility as well as the facility itself. We don't want the organism to become established in any niche spots and recontaminate the food product from the plant. Remember my example of the Salmonella agona in the cereal plant. And of course, in personal hygiene, we all remember Mary Mallon, so we don't want any typhoid Marys working on our facility. Remember, our goal is to prevent growth so that we can minimize the chance of that contamination going over the threshold of being an infectious dose. But that will not necessarily ensure product safety. One thing to remember about Salmonella, it does have a low infectious dose, and it is an infectious agent, so it actually has to get into the victim and grow within their system in order to make them ill. So we really want to keep those numbers at the absolute lowest possible. Let's talk about Escherichia coli or E. coli. Again, this also is not a spore former, commonly found in the intestinal tract of warm-blooded animals, birds, humans. Also survives freezing and dehydration and has a very low infectious dose, even lower than Salmonella. It is notorious for its ability to make people sick with just a few cells. In fact, it is sort of a cousin, microbiologically speaking, of Salmonella. The ways that we keep it in check also are very similar to those that we would use for Salmonella. Thorough cooking or pasteurization, refrigeration if it is a cold stored product, very low pH or acidity, proper hygiene for both the facility and personnel, and again, once again, as with Salmonella, preventing growth doesn't necessarily ensure product safety because of that low infectious dose. Listeria. Listeria is common in soils, so if you have products coming from the outside or you have traffic from the outside, such as forklifts or foot traffic into your facility, you have to think about Listeria possibly being tracked in. It survives freezing and dehydration very well, and one of its unusual traits is that it can grow at refrigerated temperatures, even down to 31 degrees Fahrenheit, which is pretty chilly. It is very common in produce due to cross-contamination from the soil. So this is a major risk factor if you're thinking about products like cabbage or leafy greens. How to keep this organism in check? Again, low pH is one way that we do this. We also use heat treatments to kill food products such as processed meats and other items, or milk, in fact. Pasteurization is one way to do that, or cooking. Reducing the water activity is another factor. Again, it does not grow well at low water activities. We want to avoid any kind of recontamination in the food processing environment. Listeria is a common environmental contaminant that we see in food processing facilities. Because it is so common in soil, it often gets tracked into the facility and winds up in areas like floors and drains. So we really have to have a rigorous environmental monitoring program looking for Listeria and avoiding any kind of recontamination from those sites onto the food product. Clostridium botulinum, or CBOT as it is commonly referred to by microbiologists, is a spore former. It is very resistant to heat processes. It likes anaerobic conditions. In other words, it grows without oxygen. It prefers environments where there is no oxygen. So in canned goods or vacuum-packed products where we've basically removed most of the oxygen, this is the ideal environment for those spores to germinate, grow, and produce the toxin. The spores, again, are very heat resistant, and we don't want any of this organism in our food products. It produces a dangerous neurotoxin that can actually paralyze our ability to breathe. Keeping this organism in check includes a low pH, so low acid canned foods are often some of the highest risk products we look at. Proper cooking, if it is a low acid canned food, for example, retort cooking, So if you've ever been into a cannery, they do some pretty heavy-duty cooking of those food products, and this is why, to get rid of botulism. Low water activity, low pH, and growth inhibitors are all ways to keep it in check. Otherwise lovingly known as staph. This is common in skin nasal passages, so if someone has an infected sore on their skin, for example, it is often staph that is present, so that's something that you have to think about if you have a worker with such a condition. It is very salt tolerant, not a spore former, but it produces a very heat-stable toxin. So sometimes foods can be contaminated, the organism grows, the product is cooked, but the toxin is still present. Keeping this organism in check includes things like the employee controls. For example, skin abrasions, any kind of sneezing or this sort of thing. We have to make sure that the employees are practicing very good personal hygiene, keeping their hands clean and sanitary before they touch the product. Low water activities, again, as with most of these food products, a very low water activity or availability of moisture will keep the organisms from growing. Refrigeration or hot holding, remember 40-140. We want to keep cold foods cold and hot foods hot. If we avoid that danger zone between 40 and 140 degrees, the organism is unlikely to grow and produce that toxin. So that's really what we're trying to do. We're trying to prevent growth and toxin formation with staph. Let's talk a little bit about infectious viruses and parasites. These are a little bit unusual. We've been talking a lot about the bacteria that are quite able to grow in our food products. But viruses and parasites don't typically grow in food. They're often present in the food and cause disease either because of employee cross-contamination, perhaps someone was ill with a virus and was shedding that virus and didn't practice good safe handling practices, so they were actually able to contaminate the food by handling it, or through contaminated water. We often see parasitic infections in food coming from developing nations where there isn't good sanitation, good sewage control, and so sometimes there is contamination of the water used for either washing the produce or irrigating. So those are two of the main ways in which viruses and parasites get into food products. How to keep these organisms in check? Water supply controls. If you've been through the training for the produce safety rule under FSMA, you know that there are rigorous requirements for water testing and making sure that the water supply is safe and free of any microbial or sewage contamination. Employee controls, making sure that the employees are healthy and are practicing safe hygienic handling practices, hand washing and that sort of thing. Raw material controls, if you are importing a food product, a fresh food product, we want to make sure that it was grown in such a fashion that it was not contaminated. Heat treatments also are available, so if you are thermally processing a food product, that will help in destroying these organisms. One thing to consider though, they can be very resistant to sanitizers. Some of the viruses as well as some of these parasites. The parasites produce their own structures called cysts, which are a little bit like those bacterial spores we talked about. Not quite as resistant, but still pretty tough to kill, especially with sanitizers.